Our next speaker, Chen, are you there? Great. Hi, Brian. How are you doing?
Good. How are you doing, Morton? You look younger each year.
It's
I
the
thought I was doing
it's
good for an
reprogramming,
80-year-old.
reprogramming the ARDD program.
Okay.
Brian, whenever you're ready, you can share your screen, and
All right. How does that look?
that looks fantastic. Thank you so
All
much.
right.
Go ahead.
Great. Well, I'd like to thank you, Morton, and Danielle and Alex for inviting me to speak. Obviously, you gave me the impossible task of following David Sinclair, so I'm set up for failure. But this is one of my favorite conferences every year to attend on aging. So thanks for inviting me. It's been great to see a lot of discussion around epigenetic clocks at this conference. So I switched my talk a little bit to kind of talk more broadly about predictive algorithms. So hopefully that'll be somewhat interesting to the rest of you. I'm going to talk about some of the nuances. I'm going to illustrate it through some of the work that I've been involved in, and then I'll end by proposing some guidelines to help move the field forward. I think it's still standard practice to start every talk on aging with the hallmarks of aging. I don't think I need to remind this audience that epigenetics is still one of the key molecular dimensions involved in the aging process. And David had mentioned epigenetic drift. Prior to that, also, there's been observations that there's global hypomethylation across the methylome with age. And so all of these with the epigenetic clocks, I believe, are just kind of the tip of the iceberg in understanding the role of epigenetics in aging. I think it's going to be complex and fascinating. But I'm going to focus on some epigenetic clocks right now, since that's trending. And for those who are unfamiliar, which I doubt that there are many at this point, with epigenetic clocks, they are just algorithms. And they are developed through machine learning, and you use DNA methylation data as inputs. These algorithms are usually developed through a training data set where you use to define the coefficients, which are the beta coefficients here, the beta naught, beta 1, beta 2. And then you plug and chug the methylation values or any other values for each individual into that equation, and you get a model prediction. And in this case of epigenetic clocks, that's chronological age. By definition, just as a side note, a clock measures some unit of time, but you can develop algorithms in this method to classify pretty much any variable that you're interested in, not just chronological age. And so the units can change. And so sometimes people use the term clock when it's not actually measuring time. But that's neither here nor there. And I'll just add that at this stage, as you already know, epigenetic clocks in non-human animals have been emerging in the literature since 2014. First in humpback whales, surprisingly, but then now dogs, wolves, mice, dolphins, elephants. Even a new publication came out from Steve's lab on cats just 2 days ago. So certainly not a fluke or confounding in human populations. It's a well-conserved phenomenon across multiple species. And then I'll also add, just for interest's sake, that there are a number of other uses for epigenetic clocks, not just for aging research, but in ecology. They use it to estimate the ages of animals. Turns out they don't carry around their birth certificates. This has also been used in refugee situations, for consumer infotainment, and then for forensics, obviously. I've even heard of some companies trying it in life insurance. Now, to illustrate some of the nuances that are universal to algorithmic biomarkers, which I'm defining as something that you don't see as a physical object under a microscope, but it's a mathematical manipulation of those things. And that provides us great flexibility and power. However, there are some nuances that I think are important to discuss. So in this case, I'm focused on tobacco use, which we all know is a super important contributor to health span and lifespan. Not super sexy at this stage, but I think it's pretty interesting because it's a very simple trait. In this case, is this person a habitual smoker? Yes, no. simple as that. And so just like with chronological age, you can build a highly precise algorithm to classify these individuals. So on the left, I have a very simple single epigenetic marker. This one is in the AHRR gene, which is an aryl hydrocarbon receptor repressor. That's often associated with smokers. And this is highly significant. As you can tell, the p-value is 2.2 times 10 to the minus 8. And so on most papers, association studies, you see these really astounding p-values. But if you were to look at the actual data here, if this was a diagnostic test and you got a reading of a methylation level of 80%, it's going to be difficult to say whether that person is a smoker or non-smoker based on just that information alone. And that's just because the distinction using a single marker is so low. You contrast that with what's on the right. You have a multi-marker algorithm that we developed. And here you can see super separation between the two groups. The p-value is out, as David was showing, astronomically small. The downside here is that the algorithm is really hard to interpret. So if you're interested in biology, the multi-marker algorithm as a black box may not be great, but it's fantastic for statistical discrimination between a smoker and a non-smoker, let's say. But the marker on the left, a single simple marker, could be helpful for understanding biology more so. So bottom line is, it depends. What is your goal? Are you trying to understand biology, or are you trying to classify people? The other question also is, what level of performance is good enough? And how much money and effort do you want to spend on such measurements? Is measuring just one locus sufficient, or do you want to run an entire microarray or sequence? In some cases, you do care. There's a trade-off between accuracy and convenience or costs. And so we encounter these situations where there are use cases where just measuring a handful of epigenetic markers could save a lot of time and money. But in other cases, accuracy matters. And so we, evaluating our models and algorithms, we oftentimes run a model evaluation like this, where on the far right-hand side, we start off with a full model, in this case, looking at 475 different loci. And then we successively reduce the number of loci that we use and try to see how low we can go and what the accuracy trade-off is. So I think what I'm trying to illustrate here is that there is great flexibility with algorithmic biomarkers, because you can design and tune them to perform in any way that you prefer. And that's unlike clinical biomarkers like cholesterol or CRP. You get what nature gives you, and so it performs the way it is, and you don't really manipulate it. The downside with these algorithmic biomarkers is that you have to be highly mindful and about how you're designing it. And you have to conduct the extensive testing to prove that it's actually doing what you intended. So another approach that we use to understand our algorithms is through experimental perturbation. So in the case of this tobacco test, we wanted to see if 2 weeks of quitting cold turkey would affect the epigenetic tobacco score. And so to test this, we recruited 100 participants who were all smokers, verified through biomarkers. We told them to stop smoking for 2 weeks, and then we followed them every 2 days and got their urine and saliva, so we can use the urine to make sure that they didn't restart smoking. And then we measured the epigenetic score for tobacco in the saliva and the blood for the entire 2-week period. And for simplicity, I'm just going to show you data for one subject, but the results are actually pretty strikingly consistent across all the subjects that we looked at. But the tobacco algorithm was able to correctly identify a regular smoker at any point during the entire 2-week period of not smoking. So we identified them as a habitual smoker, even though they hadn't smoked for 2 weeks. Meanwhile, other traditional biomarkers like cotinine drops off within 3 days, as you would expect based on its half-life. And in another study, we confirmed of people who quit smoking, some recently and some longer-term quitters, we showed that this specific algorithm identified recent quitters as non-smokers at around this, at least by the 60-day mark. So we know that this marker is kind of capturing a more stable measure of current smoking, similar to how HbA1c captures kind of an averaged fasting glucose level. So these are just some examples of the extent to which we go to understand our epigenetic algorithms. And for time's sake, I'm just going to summarize kind of my cheat sheet of how one should go around about validating and characterizing these algorithmic biomarkers, because there are some subtleties that sometimes we don't intend when we're developing them. I think the first thing you need to do is demonstrate that your study is scientifically reproducible. This is something that we all know. So making your data publicly available, allowing others to verify this internal validity of your experiment, I think that's first and foremost. Second, the idea of replicability is different. This is other people taking the same concept, conducting their own experiments and showing the same thing. And then the third thing is reliability. How consistent are your results within technical replicates? And does it perform as well across all levels of, let's say, smoking levels? Or are there limits of detection where it becomes unreliable? And so to expound on the idea of replicability, so verification through independent studies, I think is paramount and should never stop happening, because oftentimes there are intentional or unintentional differences between populations or different studies. And when you see a certain algorithm not working as well or working a little differently, that gives you some insights into what you're actually capturing. So I think that adds value. That's a good thing. The other thing is using corroborating evidence, such as the other hallmarks of aging or other functional measures of aging. I think that's super helpful for understanding what it is your algorithm is actually tracking with. This is helpful for designing future studies to know what is the overlap, what are some independent orthogonal measures that you can capture. And then lastly, I think, as I said before, experimental methods not only allow you to make that causal connection, but also it reveals sometimes further insights into the characteristics or the limitations or advantages of your algorithms. So I think all this sounds great. So kind of what's the problem, right? So the problem in my eyes, and I've spoken with a couple other researchers in the field, is you even with just 3 markers, let's say it's 3 loci on your epigenome, but it could be any 3 biomarkers like HDL, LDL, triglycerides, you can develop an algorithm using any combination of those. And it could predict some sort of outcome of interest like heart disease. But then somebody like your postdoc could come along and make one small little tweak. Maybe they excluded a few individuals and they come up with a new and improved algorithm, simply because it's so easy to make small changes and create new algorithms out of just a handful of markers. So imagine applying this not just to 3 markers, but to hundreds of thousands of markers, as is the case with most high dimensional data sets like epigenetics and RNA-seq and microRNA proteomics. So the problem that we might get into is you have something that starts off as a simple 3 measurement situation, but then you actually can get infinite measures out of this. And what problem does that create? Creates a little bit of confusion in the field, which is fine. But for people outside of our field who are trying to use these measures, what I get approached with oftentimes is, which one do I use? And so each one has their own subtle differences, and that creates some trouble. So these are the general guidelines that I usually my heuristic of going through and guiding people. First one is define what the goal is. What is it that you hope the said algorithm will capture? Are you interested in biological mechanism, or are you simply looking at classifying or stratifying individuals? Second is is this a statistical do you care about statistical discrimination of cases versus controls? Is accuracy important? Or would you rather have something simple and cheap, like a single marker? Other questions are, do you want to infer the information, the age, let's say, of an organism or its tissue or a single cell? I think these are fundamental design questions around an experiment. And then the last question is, who should the algorithm be developed in, and then in whom should it be used? Second, sorry about the numbering, is I always view validation and characterization of your algorithms as being paramount. And so independent validation is, of course, a must. In the genome-wide association study world, at some point they realized that there could be infinite GWASs out there. And so journals required replication cohorts. And I think we should do the same for developing new algorithms. Studies have to be reproducible, obviously, and reliable, replicable across multiple populations. I can't emphasize that enough. Depending on the use case, these algorithms may need to be highly reliable. So if we're trying to use longitudinal measurements of the same people over time, you got to make sure that any changes that you see are true and not just measurement error. And then we talked about the value of experimental validation to get at causation, and then corroborating measures that help you characterize your algorithm better. So the first kind of top part I view as dictating a priori which measures you might want to use. The second guideline dictates which measures you can trust and how you might be able to interpret your findings if you see something different. So that's a lot to consider. So for those who are overwhelmed even from that, I'll offer the super cheat sheet, which is largely based on my experience and what I've seen in the literature. A lot of people are basically limiting themselves to 4 measures. They start off with the 2 most well-established first-generation clocks. These are measuring chronological age because there's a lot of literature out there. So you want to take a look to see how that's faring in your experiment. But then there's a lot of really interesting associations coming out of the second-generation clocks, the pheno age and grim age. And they're second generation because they are measuring something other than chronological age. They're striving to get towards biological age, which is what we primarily are interested in. And the good news is that all 4 of these are free outputs from Horvath's online calculator, so it's easy for people to access. So to summarize, there are many clocks and it's counting. Outsiders want guidance on which clocks to use, but the current trend that I'm seeing in the field is that we're just building new clocks and we're applying them, which is great. But I think we need to go back and put a little bit more emphasis on studying and validating and characterizing them. So with that, I'm just going to make a plug for future collaborations. We run a lot of epigenetic microarrays. We helped develop the mouse methylation array that just came out about a year ago. And so right now we're interested in large lifestyle intervention trials that increase health span and lifespan. We're also interested in bold geroscience research seeking to elucidate the interconnection between epigenetics, of course, and multiple hallmarks of aging, and then relating that to functional outcomes. So if anybody has any great ideas they want some funding for, happy to do that. And then I want to give a shout out to our small but mighty team and Foxo Labs. We're spread out across the US, but we are highly connected and it's a highly functional team and we are always hiring. So keep an eye out. And I also want to give a shout out to our collaborators who help us at many levels, from helping to develop the mouse array to conducting some large studies that I'm hoping to talk about next year, where we're running arrays on 11,000 individuals followed for over 30 years. So more to come on that, and I'll pause there for questions.
Thank you so much, Brian. That was really interesting. So we have a question on Slack, which is, a sort of fundamental question in the epigenetic field. What is the relationship between CPGs and gene expression? Is there a relationship between the epigenetic clock and a gene expression clock?
That's a good question. I think to answer that, you would have to do like an experiment, because I think the problem in a lot of population studies, we've tried to do this extensively, and there might be some publication bias there, so just to warn you, but you can't take kind of like these are bulk cells, first of all, and you're taking them at a single snapshot, so there might not be a perfect alignment in population studies.
Okay. I have a sort of on the technical side. So, do these epigenetic clock, do they work on fixed tissues? If you have a tissue bank, for example, of PFA tissues, can you use your technology on those samples?
Yeah. So I think Illumina, not to make a plug for them, but I know that they have a way of kind of dealing with some of the fragmentation that occurs. And there's some evidence from my collaborators that say that fixed kit is actually really great for even unfixed DNA, and that actually improves the quality. So,
okay,
yes.
Great. And then finally, I would like to know for the stuff you did with smokers, when will it ever become better? The prediction, or are you just, if you ever smoked, are you doomed?
Oh, so well, there's independent of epigenetics, there's rich literature in epidemiology that shows that yes, if you quit smoking, your risk of getting mortality actually goes back to that of a never smoker. So it's never too late to quit.
Okay, I think that's a great ending message here. Thank you so much, Brian. Great talk.
All right.